Antimicrobial agents represent one of the main therapeutic tools both in human and veterinary medicine to control and treat a variety of bacterial infectious diseases. However, during the past 5 decades, the use and sometimes misuse of antimicrobials in both human and veterinary medicine has resulted in the emergence of strains of bacteria that no longer respond to antimicrobial therapy. Not only do antimicrobial resistant bacterial pathogens in animals pose a risk in terms of animal health, they also affect public health when transmitted to humans as foodborne contaminants. Thus, addressing the issue of antimicrobial resistance is one of the most urgent priorities in the fields of public health today.
Bacterial infections are treated with antibiotics. Antibiotic resistance occurs when an infection responds poorly to an antibiotic that once could treat it successfully. It's the bacteria that have become resistant to the antibiotic, not the patient.
The misuse and overuse of antibiotics, especially for viral infections like sore throat, is reducing their ability to save lives now and in the future. Up to 8 out of 10 sore throats are viral. Antibiotics only work against bacteria, making their use for sore throat often inappropriate and ineffective. Different antibiotic classes work in a variety of ways to inhibit bacteria. They can inhibit bacterial cell wall synthesis, cause damage to cell membranes, disrupt bacterial metabolism and restrict the ability of the bacteria to multiply. When exposed to a narrow-spectrum antibiotic, disease-causing bacteria should be sensitive to its effects and will therefore be killed or inhibited. Broad-spectrum antibiotics target several classes of bacteria, damaging good bacteria. This makes them unsuitable for self-limiting conditions. Antibiotic resistance develops when potentially harmful bacteria mutate in a way that reduces or eliminates the effectiveness of antibiotics. Bacteria can develop mechanisms such as stopping the antibiotic from reaching its target or modifying the target that the antibiotic works on. The more antibiotics are used, the greater the rate of resistance development and risk of resistance spreading, meaning the antibiotics will no longer work in curing bacterial diseases. Resistant bacteria can develop in any person who uses an antibiotic and can remain in the body for up to a year after a course of antibiotics. Once a bacterium develops antibiotic resistance, the genes providing resistance can then spread to other bacteria, rendering an antibiotic useless in fighting infections for everyone, not just the individual. Up to 80% of antibiotics used for respiratory tract infections are believed unnecessary and should be reserved for when they're really needed to help reduce resistance.
This happens because the bacteria acquire a mutation, a change in their DNA, that gives them a new protein as a tool to fight the antibiotic. There are many mechanisms by which this tool may work. It can prevent the antibiotic from entering the bacterial cell, pump the antibiotic out of the cell, destroy the antibiotic by enzymatic reaction, modify the antibiotic's target so it no longer binds to the drug, or give the cell a way to bypass the antibiotic's target, making the drug irrelevant. Mutations in bacterial genome occur all the time, spontaneously, but only the ones that confer a certain advantage would persist to the next generation. Let's consider a situation when a new mutation emerges and makes the bacteria resistant to a certain antibiotic. In the absence of the antibiotic, such mutation offers no advantage, and because mutations usually come with slower growth, the mutation would be diluted and eventually disappear in the following generations. On the other hand, in the presence of the antibiotic, only bacteria that carry such mutation would survive, and they would soon take over the whole bacterial population. The use of antibiotic is therefore the factor that drives the selection of antibiotic-resistant organisms. Mutations that confer antibiotic resistance can be transmitted not only vertically, from parent cells to offspring, but also horizontally from one bacterial cell to another, using mobile genetic elements such as plasmids or bacteriophages. This means a bacterial strain can share their antibiotic resistance with other bacterial strains and even with distantly related bacterial species. Horizontal transfer is a major mechanism underlying the spread of antibiotic resistance among bacterial species. These antibiotic-resistant bacteria can infect humans, animals, and spread between them through food and the environment. Antibiotic resistance is one of the biggest global health concerns. Infections by antibiotic-resistant bacteria are much harder, sometimes impossible to treat. Some bacteria, called superbugs, are resistant to most of the common antibiotics and are especially difficult to kill. Treatments for infections caused by such bacteria are costly and toxic to the patients. While the emergence of antibiotic resistance is inevitable, the process is greatly accelerated by misuse and overuse of antibiotics.
In any large population of bacteria, a few cells will be present which possess traits that enable them to survive in the presence of a noxious substance. In this case, the ability to fend off the action of the antimicrobial. Susceptible organisms, those lacking the advantageous trait, will be eliminated, leaving the remaining resistant populations behind. With long-term antimicrobial use in a given environment, the bacterial communities will change dramatically, with more resistant organisms increasing in proportion. This can result in a situation where the next time an antimicrobial is needed, it may not be effective to treat what was once an easily treatable infection. Mechanisms of antimicrobial resistance. Susceptible bacteria can acquire resistance to antimicrobials by either genetic mutation or by accepting antimicrobial resistant genes from other bacteria. This usually occurs through one of several biochemical mechanisms: mutation, destruction or inactivation, and efflux. Mutation. Mutation is a change in the DNA that can sometimes cause a change in the gene product, which is the target of the antimicrobial. When a susceptible bacterium comes into contact with a therapeutic concentration of antimicrobials like fluoroquinolones, the antimicrobial can bind to specific enzymes, in this case DNA gyrase. The DNA gyrase is an essential bacterial enzyme required for DNA replication. The end result is that fluoroquinolones block bacterial DNA replication, leading to cell death. However, when spontaneous mutations occur in specific areas of the genes encoding these enzymes, antimicrobials no longer bind efficiently. This allows the bacterium to continue DNA replication. Destruction or inactivation. Many bacteria possess genes which produce enzymes that chemically degrade or deactivate the antimicrobial, rendering them ineffective against the bacterium. Here, the antimicrobial is either degraded or modified by enzymatic activity before it can reach the target site and damage the bacterial cell. Efflux. Certain bacteria can often become resistant to antimicrobials through a mechanism known as efflux. An efflux pump is essentially a channel that actively exports antimicrobial and other compounds out of the cell. The antimicrobial enters the bacterium through a channel termed a porin, and then is pumped back out of the bacterium by the efflux pump. By actively pumping out antimicrobials, the efflux pumps prevent the intracellular accumulation necessary to exert their lethal activity inside the cell. Genetic transfer. Genetic material can be transferred between bacteria by several means, most often by conjugation, transformation, and transduction. Conjugation. Conjugation is mediated by a particular kind of circular DNA called a plasmid, which replicates independently of the chromosome. Many plasmids carry genes that confer resistance to antimicrobials. When two cells are in close proximity to each other, a hollow bridge-like structure, known as a pilus, forms between two cells. This allows a copy of the plasmid, as it is duplicated, to be transferred from one bacterium to another. This enables a susceptible bacteria to acquire resistance to a particular antimicrobial agent. Transformation. During this process, genes are transferred from one bacterium to another as naked DNA. When cells die and break apart, DNA can be released into the surrounding environment. Other bacteria in close proximity can scavenge this free-floating DNA and incorporate it into their own DNA. This DNA may contain advantageous genes, such as antimicrobial resistant genes, and benefit the recipient cell. Transduction. In this process, bacterial DNA is transferred from one bacterium to another inside a virus that infects bacteria. These viruses are called bacteriophages or phage. When a phage infects a bacterium, it essentially takes over the bacteria's genetic processes to produce more phage. During this process, bacterial DNA may inadvertently be incorporated into the new phage DNA. Upon bacterial death and lysis, or breaking apart, these new phage go on to infect other bacteria. This brings along genes from the previously infected bacterium.
To help control spread of antibiotic resistance, antibiotics must be taken correctly, only when prescribed by a healthcare professional, who should do so only when antibiotics are needed according to current guidelines. Antibiotics should not be used to promote growth or prevent diseases in healthy animals. Measures that help prevent infections also help reduce antibiotic overuse.